motherfucking robot. I'm a motherfucking robot with hands.
That's mother science.
The past decade has produced a plethora of innovation in the field of tissue engineering. Much of this progress has been realized through the research of novel biomaterials and through the articulation of cellular signaling pathways. Stroke significantly impairs an individual's quality of life. Therapies exist, however, the underlying tissue damage must be addressed. In Nature Materials, an injectable compound-loaded hydrogel was used to treat rats that had an induced stroke on one side of the brain. This hydrogel was made of hyaluronic acid and was specifically utilized for two reasons: lower levels of fibrous components in the extracellular matrix of brain tissue. Hyaluronic acid can be degraded via both an enzyme and matrix metalloproteinases. Here is a diagram of the study's timeline. The study was rigorous in its use of controls and ultimately produced some very compelling data. The study showed that in one group, the rats significantly recovered in total limb function as well as the pertinent timescales for recovery. The rats were tested on 3 behaviors to ascertain the outcome efficacy of the treatment: exploratory rearing, gait, and forepaw utilization. In feeding, the study showed significant improvement in the first 2 at 12 weeks, as well as significant improvement in fine motor control from weeks 8 to 12. Aside from the potential for treatment of stroke, this paper is specifically illuminating in the development of extracellular matrices for tissue engineering applications. Be it brain, dermal tissue, hard tissue, living tissue is comprised of cells as well as an extracellular matrix by which the cells can migrate on and adhere to. This is an integral component in our quest to fully restore damaged tissue to its native state. The article mentions earlier works utilizing VEGF in the treatment of damaged brain tissue. This brings me to my next.
Never say die.
This study utilized a unique method of delivery: small molecules in a polymer extracellular matrix with degradable crosslinks. They used high-density clusters of VEGF and bound them to nanoparticles made of heparin. This ultimately led to the revascularization of the tissue. The researchers discerned that the revascularization of the tissue alone did not result in the desirable amount of axonal infiltration to the damaged tissue. In strokes, microglial cells and astrocytes form a barrier around the affected tissue, preventing migration of new healthy components back into the area. They found that heparin in its unbound state helped modulate inflammatory signaling and led to a significant increase in the repopulation of damaged areas with neuroblast cells and axonal connection. The researchers analyzed regulation of inflammatory signal. They found that heparin specifically reduced activity of TNF, or tumor necrosis factor alpha, hypothesizing the binding as a mechanism. These findings are pertinent to all applications in tissue engineering. Unintended inflammatory signaling is a major culprit in poor implant outcomes, autoimmune disorders, as well as a mechanism in the previous episode over cosmic rays in the brain.
I'm a motherfucking robot. I'm a motherfucking robot. I'm a motherfucking robot.